Thank you. Because I wanted to remind, thank you. I wanted to remind that cells which circulate may undergo epithelial to mesenchymal transition, and this epithelial to mesenchymal transition is, of course, a loss of a progressive loss of epithelial antigens, and cells acquire mesenchymal antigens, and this is associated to the differentiation and to a more stem character, which is also associated to a higher potential of giving metastasis. And the other issue that I would like to point out is that we have several rare cells in blood, not only circulating tumor cells, and by definition rare cells are cells which are in blood in a range of 1 to few per ml of blood, which means one or few mixed to approximately 5 to 10 million leukocytes and 5 billion of erythrocytes. So this is the reason why we have this technical challenge that Dr. Pinzani pointed out. The different circulating or rare cells we have in blood are tumor cells, including different type, epithelial tumor cells, mesenchymal tumor cells, tumor stem cells, circulating tumor microemboli, but also non-tumor cells like epithelial normal cells, they are in blood, epithelial atypical cells, endothelial cells, stem cells, and in pregnant women, fetal cells. So we need to distinguish all those cells, and of course the technique we apply to isolate them may or may not introduce bias. So our team developed ISET, and I was pleased to hear Professor Pinzani explaining the beginning, because in fact this technique has been developed in our team, and with Giovanna Vona, that unfortunately disappeared, who was coming from the team of Dr. Pinzani. This system has one characteristic, is that the system does not lose cells, in fact. It's true, it seems magic, but we validate all type of filters and buffer, and you can come to the lab, we put one cell and we can recover it. One cell in 10 milliliters of blood. So how does it work? Very simply, 10 milliliters of blood are diluted with a proprietary buffer and then let to stay 10 minutes to lysate the red blood cells and fixate the cells. And then the blood is charged in a consumable, which is a cartridge containing the filter, and after a few minutes the filter are reduced to the filter and then can be analyzed by morphological analysis, molecular analysis, so all types of analysis. This system is generally used with fixed cells, but can also be used now because the protocol has been developed with fresh unfixed cells without losing cells. Okay, so what also I have to point out that as I said, in blood we have different type of cells, and we need to distinguish the one from the other ones. So a group of pathologists has published, as you can see here, the group of Professor Hoffman, that in patients with cancer we have benign epithelial cells circulating, atypical but not really, frankly, tumor cells, and tumor cells that they recognize by cytopathological analysis, thus using the classical cytopathological criteria which are indicated here. We just thought to combine the molecular analysis at the single cell level with the cytomorphological analysis to see if molecular analysis could help the cytomorphological identification of circulating tumor cells. In a particular case of tumor, the clear cell renal carcinoma cancer, where the cytopathological identification of cells is particularly difficult. And this cancer has also another advantage from the biological point of view, is that the majority of these cancers are characterized by the mutation in the von Hippel-Lindau gene, and this mutation is found as a heterozygous mutation in 84 to 98% percentage of cases, and has homozygous mutation in 50% of those cases. Although we don't know which is the significance of this mutation in the cancer, the gene is a tumor suppressor gene. We don't know if this is the diagnostic mutation or just a tumor initiator or a mutation which is a step in the tumorigenesis. We thought to use it to compare the presence of mutation versus the cytopathological diagnosis of these cells. Thus, we tested 30 patients with sporadic clear cell renal carcinoma. Only 2 had metastasis. The filters were stored in 2005, and they were analyzed between 2012 and 2013. So we still had the DNA available for this single cell analysis. We analyzed circulating tumor cells and circulating atypical cells and compared the result with the result we obtained from the tumor tissue and the genomic leukocyte DNA from the same patients. The workflow is here. The cells were isolated by ISET. They were stained, cytopathological diagnosed, and then laser microdissected. And after lysis and whole genomic amplification, we applied 5 PCR covering the 3 exons, and then the PCR products were sent for sequencing. We found 18 different VHL mutation, and in 22 patients and 119 cells we found 1 only mutation, but in 3 patients we found 2 different mutations in 19 cells. No VHL mutation was found in 4 patients and 23 cells coming from those 4 patients, but these patients had a tumor without VHL mutation, so we could not conclude. Among the 30 patients which were studied before surgery, 29 had CTCs and or CAC. 20 patients had both circulating tumor cells and circulating atypical cells, and 9 only circulating atypical cells. Furthermore, 25 of the 29 had the VHL mutation in the tumor, which means that 4 patients had no VHL mutation in the tumor, so we could not really conclude in these patients. This is just a compilation of the mutations that we found. We found transversion, transitions, and friendship mutations. Here are some analysis. You can see circulating tumor cells. You can see that we found this transversion in codon 9, exon 1 in the tumor cells, and the same in the tumor tissue as compared to the genomic DNA. Again, an example. Here we have a mutation that we found in CTCs from 2 patients, as you can see here. The mutation in the CTC is a transversion in codon 88, the same we found in the tumor tissue, and of course this is different from the genomic DNA. Now, concerning the cytopathological versus the molecular data, this is a sort of global results analysis. We microdissected 327 cells. We got WGA, whole genomic amplification signal from only 2005, and of course this allowed us to have further molecular analysis. Among those cells, those single cells, we had 57 circulating tumor cells, where we found the mutation, which was the same mutation we found in the tumor tissue, and we also had 7 circulating tumor cells where we didn't find mutation. So these 7 cells were coming from tumors, as I said, without the VHL mutation, so no conclusion on that. And then we also analyzed and found those 104 circulating atypical cells. And among these, we found the same mutation that we found in the tumor in this 104, And in 21, we found a VHL mutation which was different from the one that we found in the tumorous tissue, raising the issue that could be a variant clone. And in 16, circulating atypical cells, we didn't find mutation, so again, no conclusion. So overall, we had 57 circulating tumor cells and 125 cells with VHL mutation. So to wrap up, at the patient level, we studied 30 patients and found CTC in 20 or CSC in 29. 20 patients with both CTC and atypical cells, and 9 only with atypical cells, and 25 of those patients with the VHL mutation in the tumor. 4 patients showed no VHL mutation in the tumor, nor in the CTCs and in the atypical cells. At the cellular level, 20 patients we found 20 patients with CTCs having the same VHL mutation than in the tumor tissue, and 25 with CTC and or atypical cells having the same VHL mutation than in the tumorous tissue. So, if you consider VHL as a diagnostic mutation for the identification of tumor cells, we can say that the cytopathology in this study had 100% specificity and 80% sensitivity. However, we need to consider in the use, thinking of the use of this molecular analysis for cytopathological diagnosis, cost-benefit consideration, and also the fact that we do not know the real significance of the VHL mutation, so we don't know if we can use it as a diagnostic significance for the cytopathological molecular diagnosis of the presence of CTCs. And finally, I would like to thank all those who participated in this study, Dr. Benjamin, who did all the molecular study, the pathologist in Nice, Professor Hoffman, Dr. Veronique Hoffman, and Dr. Ili, the pathologist in the University of Tunis, Professor Chabouni, Dogri, and Robnan, and Professor Mejan, who is the urologist in Hôpital Georges Pompidou. And thank you very much for your attention.
Thank you. One question? No? Thank you.